Translation is the second step in the two-step process by which DNA directs the synthesis of proteins. Once an mRNA molecule moves out of the cell's nucleus and into the cytoplasm, the translation process begins. In translation, the information carried by the mRNA is read, and ingredients present in the cytoplasm are used to produce a protein. First, there must be large numbers of free amino acids floating around. Amino acids are the raw materials for building proteins, and an essential component of our diet. Second, there must be molecules called ribosomal subunits, the protein production factories where amino acids are linked together in the proper order to produce a protein. Finally, molecules called transfer RNA or tRNA interpret the mRNA code and link specific base sequences on the mRNA with specific amino acids to make proteins. tRNAs play a central role in translation. tRNAs are two-sided molecules. On one side, a particular amino acid is attached. There are different tRNA molecules for each of the 20 different amino acid molecules, which are floating around in the cytoplasm in great numbers. On the other side of a two-sided tRNA molecule is an attachment site, consisting of a 3-base sequence. The attachment site matches up with a 3-base sequence called a codon on the mRNA transcript. Note that only a particular tRNA will match up under proper base pairing rules with a particular codon. An A for adenine pairs with a U for uracil, and a C for cytosine pairs with a G for guanine. Thus, the tRNA translates the code on the mRNA into an amino acid that will be used to build a protein. Translation occurs in 3 steps, the first of which involves recognizing a start sequence and initiating protein building. Recall from the transcription of a gene that the mRNA acquired a cap at one end. In the first step of translation, the cap allows the small ribosomal subunit and a special initiator tRNA to bind to the mRNA and scan for the start sequence on the mRNA transcript. The initiator tRNA uses base pairing rules to bind to this sequence, which is always AUG. The initiator tRNA has the amino acid methionine bound to its other side. This will be the first amino acid in the protein that is to be produced. In the second step, the ribosome elongates the amino acid sequence to make a protein. A tRNA carrying a different amino acid recognizes the next 3 bases on the mRNA. The ribosome then facilitates the connection of this second amino acid to the first. The mRNA threads through the ribosome by 3 base chunks. The free tRNA floats away, and another tRNA decodes the next codon. As this process continues, the protein grows. All proteins are chains of amino acids, like beads on a string. The last step is termination, which occurs when the ribosome encounters a 3-base stop sequence on the mRNA, which signals the end of translation. The amino acid strand and mRNA molecule are now released from the ribosome. Depending on how long the mRNA lasts in the cell, the same mRNA strand may be translated hundreds of times. Eventually, it is broken down by enzymes in the cytoplasm. Through the 2-step process of transcription and translation, an organism converts the information held in the DNA of its genes into mRNAs, and then into the proteins necessary for life. But the process is only as good as the organism's underlying genetic information. Sometimes, something occurs to alter the sequence of bases in an organism's DNA. Such an alteration is called a mutation. A mutation is copied along with the rest of the sequence into the mRNA during transcription. A new 3-base sequence in the mRNA typically results in a different amino acid being incorporated during translation. This type of mutation is called base pair substitution. Insertions or deletions, as in this example, can be much more harmful than substitutions. If a single base is added or removed, the 3-base groupings get thrown off. During transcription, this new arrangement is copied into the mRNA. Consequently, the sequence of amino acids stipulated downstream from that point will be all wrong. The reading frame is shifted. Mutations can have a range of effects. Sometimes the results are serious, even deadly. Other times there is little or no detrimental effect, and occasionally, but rarely, the mutation may be beneficial to the organism. Why do many Asians have unpleasant experiences associated with alcohol consumption? Some people respond to alcohol with a fast flush response. At a dinner party, before Isabella finishes her first glass of wine, her face turns crimson, her heart begins to race, her head starts to pound, and she soon feels the need to vomit. In contrast, although her friends become tipsy, they otherwise feel fine. How can people respond so differently to alcohol? The answer lies in a particular gene in the DNA. Certain mutations in this gene result in fast flush responses. The normal gene and the mutated gene differ at a single base in the DNA sequence. This difference is transcribed into the mRNA and translated into the protein. While the normal sequence results in a normal enzyme, the mutated sequence results in a non-functional enzyme. Consider how the body breaks down alcohol. It's a two-step process involving two different enzymes. The enzyme we've been talking about, called aldehyde dehydrogenase, is enzyme 2. In the first step, the first enzyme converts molecules of alcohol into molecules of another toxic chemical. In the second step, the second enzyme converts these toxic chemicals into non-toxic products. In people carrying the mutated gene, the non-functioning enzyme cannot eliminate the toxic chemical. This chemical builds up and results in symptoms of the fast flush reaction. Approximately half of the people living in Asia carry a non-functional form of the gene for aldehyde dehydrogenase, a mutation that may actually confer a greater benefit than harm. In a study of 1,300 alcoholics in Japan, not a single one was a fast flusher, even though half of all Japanese people are fast flushers.